Good afternoon, everyone. I'm Tyler Jacks. I'm president of Breakthrough Cancer, and I'd like to welcome you to the latest episode of Breaking Through, which is a webinar series that we designed to bring you insights from the front lines of cancer research and treatment. My guest today is Mark Goldsmith, who is the CEO of Revolution Medicines. Mark, welcome. Thanks for being here.
Thanks, Tyler. It's really a great pleasure to be with you today.
I'm very much looking forward to our discussion. Before we jump into that discussion, let me say a few words about Breakthrough Cancer and sort of set the stage. Breakthrough Cancer is a 5-year-old cancer research foundation that was built to support interinstitutional team-based cancer research focused on very difficult, long-standing problems in cancer. In this organization, we practice what we call radical collaboration, which brings together researchers and institutions from across the country in a team-based model, highly functional teams, to bring the best of cancer science to the cancer clinic so as to accelerate progress and improve outcomes for patients. Radical collaboration isn't only about the scientists coming together or their institutions coming together. It's about partnership at all levels. The teams that we invest in partner importantly with industry, which are developing the emerging powerful drugs to treat the disease. We also work closely with other funders who, in a similar fashion, believe in this team-based approach. And importantly, we partner with patients who are critical to this process of learning in patients, learning as much as we can in the clinical setting so as to go faster and ultimately do better. And the project that we're going to talk about today is a perfect example of radical collaboration in action. It brings in all of those elements, as will become clear as we talk about today's story. So I want to begin, Mark, in reflecting on the clinical trial results that your company revealed quite recently. Revolution Medicines is a 12-year-old biotechnology company that has, among other things, developed powerful medicines that target a class of proteins called RAS proteins. These proteins are activated in a wide range of cancers, particularly widely in pancreas cancer, and they're believed to be, known to be, important drivers of the disease. Mark, last month at the American Society of Clinical Oncology annual meeting, results from your Resolute 302 study were presented in a plenary session. The study tested the effects of daraxonrasib, your newly developed inhibitor of RAS proteins in pancreas cancer in patients who had failed frontline therapy. When Brian Wolpin from the Dana-Farber Cancer Institute presented the critical slide, thousands of people stood up and applauded. A spontaneous standing ovation, which I can tell the audience is not common at a scientific meeting. So, Mark, I'm curious, what did you report that day? And talk a bit about the emotion that you and your colleagues felt at that moment.
Well, thanks for the great introduction. It was quite an experience. As you mentioned, we reported the results of a roughly 500 patient randomized controlled global phase 3 clinical trial. It compared daraxonrasib to standard of care treatment for second-line pancreatic cancer. Standard treatment basically consists of chemotherapy. In fact, in almost all lines of pancreatic cancer, chemotherapy is the backbone. And physicians could choose amongst 5-FU-based or gemcitabine-based standard regimens, if the patient was randomized to chemotherapy. If not, if they were randomized to daraxonrasib, they received 300 milligrams of daraxonrasib daily. That's a once daily dosing. And the results were really, quite astonishing. The overall survival seen in the overall intent to treat population showed a 60% reduction in the probability of death. That translated into a median overall survival of 13 plus months compared to the control group, which had a median overall survival of 6.7 months, very much in line with what we expected based on historical experience. Likewise, the progression-free survival showed a hazard ratio of 0.49, just under 0.5. The overall response rate was 32%, about 3 times that for chemotherapy. And importantly, patients also reported preservation of quality of life during their course of treatment compared to chemotherapy. And interestingly, that portion also had a hazard ratio of about zero point 0.5 in the 0.5 to 0.6 range. And so all of these different parameters together added up to a remarkable overall experience for patients, both increasing their lifetime, increasing their time without progression, and stabilizing quality of life, which are, the pillars of what patients are looking for. It was generally well tolerated, as we had seen in the phase 1/2 trials. A very small percentage, roughly 1%, actually discontinued drug because of side effects, which is quite a low number for an oncolytic therapy. But there are some common side effects, and those were reported again in these trials that we had seen before. The most common side effects are skin rash and gastrointestinal side effects, which can be either in the lower GI tract causing diarrhea or in the upper GI tract causing stomatitis. These are generally well managed and were generally well managed in the trial, typically grade 1 or grade 2, occasionally more severe than that. But usually when there's something more severe like an extreme rash, that can be associated with sun exposure, so things that are outside of the actual treatment regimen, or some sort of failure in the prophylaxis regimens or lack of treatment when it does occur. But as I said, coming off of drug entirely was very uncommon, although there were some dose reductions. Overall, a good experience for patients.
Yeah. I want you to talk about the emotion.
Mm.
I wasn't there at the meeting, but I've seen videos. I saw not just clapping, but literally tears in people's eyes.
Yeah.
And I imagine that might be a sentiment that was shared in the halls of Revolution Medicines as well. This was a moment that we in the field have been waiting a long time for. Just talk about it, how it felt.
Well, you're bringing back all those emotions by asking about it. It was and remains a very intense emotional experience. Partly because we've committed so many years to trying to achieve a breakthrough, but also because of what it reflects. I mean, these results launched daraxonrasib and RevMed onto the global stage instantly. It was astounding. It's still astounding to us, the scale of the reach across the globe. And I think that is indicative of what underlies all of this effort, which is a hunger, a need for new treatments that change the outcomes. We just haven't had that in pancreatic cancer. And now we're finally addressing the root cause, the underlying cause. I know we'll talk about that some more, but that gives us a real chance to make a difference. So, we are very passionate about what we do. The patient mission is very much in our hearts. But I also know you appreciate that the science is in our DNA. It's something that we really are deeply committed to, and we think it's the only way to achieve unprecedented results is through science.
Yeah, it's a wonderful example of committing to understanding the science and then modifying the relevant driver in the disease process, and then fortunately in this case having a major impact. We're not done. We'll talk about that as well, but it's a
Yeah.
major step forward, and all of us in the field feel strongly that way. So we share in the excitement and optimism for the future that I know that you all feel as well. But I want to back up a little bit here. You and I first met each other in the summer of 1983
Mm.
as we arrived on the campus of the University of California in San Francisco to begin our training. Me in science, you in science and medicine. And you'll remember that in the previous year, in 1982, 3 laboratories reported for the first time that RAS genes were mutated, activated in human cancer. It was actually the first description of the first human cancer genes. 1982. We fast forward 44 years later. You're the CEO of a high-flying company. that's made the most powerful RAS targeting drug in pancreas cancer. I've been studying RAS and pancreas cancer for 25 plus years. Um, it's a reasonable question. 44 years is a long time. What took so long?
Well, I have to tell you, in 1983, I did not know that the year before RAS was discovered as the first human oncogene. I only appreciated that in recent years. Nor did I ever imagine that I would be sitting here talking with you 43 years later about targeted therapy for pancreatic cancer. So it's quite a crazy and interesting story. Um, People have been trying to drug RAS for a very, very long time, as you know, decades of effort. Um, there are no obvious druggable pockets on the surface of RAS the way there are for some other drug targets, receptor tyrosine kinases, and so on. Um, and so high throughput screening kinds of strategies that have been typical in the pharmaceutical industry for the last 25 years just didn't cut it. One couldn't get high enough affinity binders. Um, and so that sort of led to a reliance on chemotherapy, fully in pancreatic cancer and other diseases that are driven by RAS. I think there have been some breakthroughs that have changed our thinking about RAS as a druggable into becoming a druggable target. No doubt that Kayvon Shokat and John Ostrom's work, in which they use covalent tethering, something that had been around in research labs for a long time, but they figured out a way to go after a really important disease target, KRAS G12C, using the cysteine as a reactive side chain, and plugged something into it that then pulled it into a pocket that sort of formed around it. And that was really the first signal that it might be possible to drug RAS. And of course, out of that first compound or series of compounds became the prototype that became the industry standard for a few years, which is the KRAS G12C off inhibitors, as you know. That was a major breakthrough and provided ultimately the first evidence from sotorasib in humans that inhibiting RAS could shrink a RAS-driven cancer, even though you had shown and many others had shown in various model systems that that should be true based on knockout experiments and so on, genetic manipulation. But now you can see it in a human. So that's one breakthrough. Um, uh, I think a second breakthrough was the recognition that maybe we don't need to use just rule of five molecules, which is what the pharma industry has been focused on. Maybe there are other ways to go about it. And here I'd say Greg Verdine's vision, his recognition that maybe we can use a larger surface area, borrow something that nature has done with rapamycin and cyclosporin, natural products that are human drugs now, but they didn't start out as human drugs. They were found in dirt. And could we replicate that uh, larger surface area contact and use something more like a glue rather than filling a hole? Um, I don't prefer the word glue. I think it's more like Velcro, where you have lots of little, low, small contacts that are not sufficient, but when you add them all up, you get a nice tight binder. But nonetheless, to use the concept of ternary complexes, where there are two proteins involved. One's the target you're trying to inhibit. The other is a presenter protein that presents the small molecule, and the small molecule gets sandwiched between them and through a very large surface area can create high affinity and high selectivity binding. So Greg had this idea that you could do this synthetically, that you could engineer these, not just find them in nature, but engineer them, and create a company called Warp Drive Bio. That company showed some early progress. It wasn't enough to have a drug, but it was encouraging, and we decided to incorporate that technology into RevMed with their help. Um, and then built really an industrial strength platform out of that has taken us in every dimension of RAS. But I think those are the sorts of breakthroughs. Now, maybe there's one other that's important, and that is the recognition that you don't necessarily have to start with a pre-existing drug pocket to target something. Proteins fold and unfold, they breathe, they reshape themselves, and there are intermediate states where there might be a transient pocket that you might be able to shove something into. and that has created even newer small molecule compounds that might be useful in either RAS targeting or other undruggable targets. So a lot
Yeah.
of things have happened over the last few decades that have made this possible, and we like to acknowledge that we stand on the shoulders of giants for
Yeah.
anything that happens today.
Yeah. Certainly many contributors along the path in cancer genetics. And also, I would say, importantly underscore the importance of creativity and chemistry, which enabled a lot of this progress that we've seen. And now, following Kayvon and John's work and the approvals in the first phase of the RAS inhibitors, the RAS inhibitor space is probably the most crowded and active area in cancer therapeutics today. Daraxonrasib is a bit different in terms of not just the way the drug works, but also what it targets. It's not a quote unquote allele specific inhibitor or even a KRAS specific inhibitor. Talk a bit more about the attributes of daraxonrasib and why that might be important as we think about its activity.
Yeah, I think there are two major distinctions for daraxonrasib from most of the rest of the field. I think the first is it is a compound that binds to the on or activated state of RAS, which it probably logically should have been the starting point with RAS inhibitors, but it was even harder to drug RAS on proteins than RAS off proteins, and so Kayvon and John, fell into RAS off G12C. Um, but daraxonrasib with essentially all of our tricomplex inhibitors bind to the on or activated GTP bound state, which is the truly oncogenic form of the protein. The other state is simply an inactive sort of reserve pool. So that's one distinction. And I think the second distinction that you just alluded to is that it binds to all forms of RAS, really all isoforms, K, N, and H RAS, and essentially every oncogenic, natural oncogenic mutant form that we've found, it binds to it. Now, I should point out, we didn't really set out to create that molecule, although the chemistry group might have a different view of that. But we, as a corporate level approach, we're actually looking for a particular allele specific compound, which was hard to achieve. But in the process, the team created this multi-RAS inhibitor and presented it to us. And in fact, when they first presented it, didn't really point out that it was a multi-RAS inhibitor. They showed us how it was active on the particular allele we were interested in. We got excited about it, and then at the end said, "Oh, by the way, it hits every other form of RAS too." And we all sat there sort of stunned, and we weren't sure whether to applaud or to send them back to the bench. We didn't quite know what to do with that. Um, because we knew from genetic experiments, if you knock out all RAS, it's embryonic lethal, but we did study it, and I think that was one of the special features of the team here at RevMed, that they didn't just assume based on the genetic experiments that that's how a pharmacologic action would work, and put it into some mice at reasonable doses, got reasonable exposures, and didn't see much of an effect other than shrinking tumors. And that occurred across many different mutant forms and many different tumor types, and ultimately led to very large mouse clinical trials that reproducibly showed we could shrink tumors without really causing much impact for the animals. And that was really stunning to us. And we decided on that basis to take it into the clinic and to see what would happen. But there was a moment of trepidation when we started dosing
Yeah.
patients, that's for sure.
Yeah. I'm convinced that that's true. Having been one of the labs that showed that knocking out just KRAS alone actually led to embryonic lethality. So
Yeah.
I'm not sure I would have bet on it causing benefit without significant risk, but fortunately that's the way it turned out. And I give the company a lot of credit for, being bold in this regard. I also give the company a lot of credit for sort of science forward thinking. It's a very science driven company. I know many members of your team, excellent scientists, and they really follow the science and they pay attention to what the data are telling them. And this was good intuition as well, that this was a drug worth banking on. And obviously it has had quite dramatic effects. Your company, beyond daraxonrasib, is also following the allele-specific inhibitor path. Do you want to say a bit more about the portfolio?
Yeah, well, again, back to sort of a really important concept. We try to be humble, and we don't actually know, and certainly did not know 5, 6 years ago, which approach would be the superior approach, or whether they simply would be complementary. And so we decided to pursue both the RAS multi-inhibitor and to try to create mutant selective inhibitors. Now, historically, the first thing that we got from Warp Drive Bio when we acquired the technology was a collection of a few hundred compounds, all of which were targeted to the G12C allele. And when we asked various advisors, did they think this platform could be extended to other mutants? We got sort of mixed answers. Some were very gung-ho, some said no way. There was just no way to know. And so when we acquired it, we created a series of teams internally. I mean, just to show you the seriousness with which we bet on this, we bet essentially our entire future as a company. A series of teams, each of which had an assigned allele, one of which delivered daraxonrasib, the allele nonspecific, but others to work on different alleles. And of course, G12C was one of them, top priority, because we had a few hundred compounds to begin with. We also prioritized G13C, one of the rarest oncogenic forms, but it was a near neighbor of G12C. We thought if you could drug G12C, maybe you could just move it a few angstroms to the right and you could drug G13C. So from a probability of success point of view, it would be higher, even though the overall clinical impact might be lower, public health impact. But then we worked on other targets, G12D, et cetera. And all of these groups were organized around essentially a hub, a data sharing hub, where the SAR, pharmacology, synthetic routes, biology, all of that information would be poured in from every group into this shared hub. And so every other group benefited from all of those insights, and rapid advances in one group would rapidly be transferred right across to every other group. And to our great delight, and obviously to the benefit of our shareholders, but most importantly to patients, we were able to drug every form of RAS. And so this platform that started out as a G12C platform, I'm sure it was envisioned to be more than that, but it started out as a G12C platform, became really an all RAS platform. And we've been able to go beyond that now and not just use one particular approach, but to put different pieces together, almost a plug and play strategy. One of the most interesting examples was targeting G12D. Now, aspartic acid has never been targeted successfully by a drug discovery program. And that's because if you're trying to do it covalently, the hydroxyl group is highly reactive, but so is water. And so the possibility that you could ever get selectivity out of it was very low. But what our team did in tackling this, understanding that risk, was to use the tri-complex approach to first create a non-covalent interaction between the molecule and RAS G12D, and to use it to engineer very carefully the orientation of the warhead that they placed onto our molecule so that it would be just in the right orientation for that one aspartic acid. And they dialed down the reactivity of that warhead so it would only react if it was held in position long enough to achieve the reaction. So it went the opposite direction of the way chemistry had been for years, which is to try to dial up the reactivity. They went the other direction, dialed it back down, used the non-covalent binding to position it, and then achieved a covalent binder of aspartic acid on RAS G12D and very selective for RAS G12D. It doesn't work on RAS G13D, just to make that point. And we really enjoyed sitting in the audience at a major scientific conference when a speaker stood up and said, covalent approaches to drugging KRAS G12D will be impossible because it's impossible to drug aspartic acid. We already had the results. We knew we had the compound, and we just sort of looked at each other and said, well, there's another piece of dogma that's about to go down the toilet. So it was pretty fantastic. But a continuous learning cycle, I like to call it a virtuous cycle of learning, where data not only from the chemistry lab and the biology lab and pharmacology lab, but also from the clinic and translational work all get fed into the project such that we continuously are trying to improve on what we've done already. And there are no barriers that we consider insurmountable. If we want to achieve something, somebody will go give it a try and push the boundary if possible. And
Right.
that's a, that's about as exciting as you can get in science.
Yeah, incredibly exciting. For those in the audience who aren't aware, pancreas cancer, something like 90 to 92% of patients have RAS mutations, and it's a range of different ones. The one that Mark was just describing, G12D, happens to be the most common. So we can imagine that allele-specific inhibitors like G12D will have a place, either alone or in combination, including potentially in combination with daraxonrasib. I'm sure that's of interest to your team. I want to make a switch, Mark, and start to talk about the collaboration that exists between Revolution Medicines and Breakthrough Cancer. As I do, I'll tell the audience that we're welcoming your questions. If you want to type questions into the chat, we're monitoring those questions. And at the end, before we wrap up, I'll ask Mark some of your questions along the path. So now, a few years back, Breakthrough Cancer approached Revolution Medicines on behalf of a team lab that we had funded, whose mission was to target KRAS in pancreas cancer. And they had been doing a lot of work getting ready for clinical studies. And the company was developing daraxonrasib, and the data was beginning to come out from phase 1 and phase 2. We hadn't yet heard about the phase 3 by the time the partnership was discussed, although it was ongoing. The team, which includes, by the way, Brian Wolpin, whom I mentioned previously, but a large group of talented investigators, again, from major cancer centers across the country, said, we want to work with RevMed. We want to use their drug in a clinical study where we can apply the best of science to understand what's really happening when these patients get treated. The team and we at Breakthrough Cancer approached the company. We weren't certain what the response from the company would be because we knew that the company's drug was their lead asset, was in phase 3. Most companies, especially smaller biotech-sized companies, would say, don't talk to us. We're very focused. Leave us alone. Let's wait until we see the results from phase 3. Your company, by contrast, was very encouraging and actually agreed to create a cohort in an existing phase 1, phase 2 trial that would allow our investigators to study pre-treatment biopsies, on-treatment biopsies, and if necessary, at relapse biopsies with the best available molecular profiling tools and the best computational tools to actually understand what was happening in the cancer cells when they were being treated with daraxonrasib, what was changing in the tumor microenvironment in the setting of treatment. What clues might we be able to suss out that might one day lead to resistance to the drug's action? And what could we ultimately do about that as we thought about combination studies? I again give your company tremendous credit for taking that step. And I'm curious, what was in your mind that led you to say yes to this trial?
Well, I think several things. One is, as I said, I think doing science, I said it's in our DNA. I think we just fundamentally believe that if you're going to develop new treatments for patients, particularly in difficult diseases, and not just copy other people, if you're really going to do innovation, you have to understand the underlying science. And if you're not going to bother doing that, you shouldn't bother trying to develop new drugs. I mean, it just doesn't make any sense. So that's at a core. Our first two corporate values are tireless commitment to patients and transformative science, numbers 1 and 2 in that order. So that's where we start. I think we also recognize that pancreatic cancer has been deeply studied in preclinical systems. You're a leader in that space. That's really informed a lot of what we might expect to see in the clinic. But there's been very little real clinical research with meaningful lessons coming out of it. And I think that's largely been because there haven't been the tools to try to modulate a pancreatic cancer. There's just been basically chemotherapy. So we sort of feel a responsibility with a compound like daraxonrasib, if it's going to have this kind of impact, which we now clearly know from thousands of patients of experience, to understand how it works. Not so much because it's going to advance the compound in the near term towards an approval or commercial, launch, et cetera, but because it's going to teach us fundamental things that will inform us about how to do even better than that. Whether it's going to inform how to improve the molecules, whether it's going to inform how to dose the molecules, how to combine them, what sequence to use them in. There's just so many different things that we can learn. And then, as you point out, the holy grail of all of this is to know, be able to predict and have something uh, to handle resistance. And that resistance really is going to be best defined to some degree by looking in vivo at real live, human tumors. Um, we also recognize that while we have a lot of insight, we have a lot of rigorous science that goes on here, we don't have it all. And there are many folks who have created like fantastically important careers developing very sophisticated approaches and insights. And we want to tap into that. So we gave the compound to various collaborators to work on, but this was one where the opportunity was to jointly, together, actively collaborate and bring together our knowledge with this group's knowledge, our resources with this group's resources, and then, of course, our access to patients and compounds, in particular, daraxonrasib.
Yeah.
And what an incredible opportunity. And kudos to you and Breakthrough for proposing this, to all of the collaborators for signing up to it, and then to our folks who first heard about it and came in and said this is something we have to do. And I wouldn't say there was any controversy around it. It was more about how to do it and what to do rather than whether to do. And I think we spent a fair amount of time working together to design
Yeah.
design this study.
Yeah. Yeah. Let me say a bit more about it. I mentioned in the intro that this was a great example of radical collaboration. The scientists and their institutions, the company and your scientists, Malika Singh, Clay Gustafson, many others contributed greatly to the design, literally the design of the trial, what science we were going to do, how we were going to do it. They are partners in this project. They sit with the team and analyze data in real time. We also have a great partnership with the Lustgarten Foundation, a co-funder of this activity. And I mentioned the patients. In this case, the patients provide pre-treatment biopsies. That's a tissue biopsy from a tumor. On treatment biopsy, they're on the drug for a couple of weeks. There's another biopsy taken, and then if the tumor relapses, then another biopsy will be taken. The patient as partner, critically important. I'm sure at one level the patient is motivated to help learn about what's happening in themselves, but I also know from a lot of experience that they're motivated to help the field learn, to help the next patient who might be faced with this same diagnosis. So a wonderful example of radical collaboration. In my view, Mark, this project that we're doing together fills a gap in early drug development, especially with a promising agent like daraxonrasib. Early stage clinical trials, learning in patients. Talk about this as a model for your company, but maybe for the field in general. As a CEO from the industry side, how do you think about this as sort of setting a standard, if you will, for how promising agents should be investigated in the clinical setting?
I definitely think that this collaboration is setting a very high bar. Observing it the way I get to, don't get my hands wet, but I get to see and hear about it and talk to people who are involved in it, and it's really a remarkable collaboration. And I think if others can replicate that, then they're going to advance science by doing so. I don't know how broadly and easily it's going to be replicated. It takes real conviction. It takes a deep investment. You all and the Lustgarten are putting in substantial capital. We have matched that with substantial capital. Um, and I do think that clinical development activities will almost always deliver more quickly the urgent kind of near-term regulatory milestones that are needed for an approval. So I don't think we should get confused about what this is for. And I know you're not, but I think people who are hearing should hear from us clearly. This is about uncovering fundamental scientific answers to key questions about the biology of the cancer, the biology of resistance, the biology of histologic transformation and adaptation that occurs inside a tumor, helping us understand why do tumors take so much time to shrink? Why don't they shrink instantly? What's left after a tumor is getting crushed by a drug? How does the tumor microenvironment change? I mean, there's just so many things, but it's a very demanding study. And it's amazing to me how many biopsies were successfully collected in the first phase of this and successfully studied by this multi-dimensional platform. Um, it's really impressive. I don't know how easily others are going to reproduce it. I hope they do. We
Yeah.
certainly are interested in doing more of it. But again, it comes back to sort of our view that we're here not to copy other people. We're here to sort of forge new paths forward. And others might copy us. Okay. Maybe people will be stimulated to come up with their own innovation. But for our mission, we can't accomplish it without doing this sort of thing. And so we're really, really proud
Yeah.
and
Yeah.
take this very, very seriously.
Well, I think you should, but we're quite proud of it too. The data are coming in. We're not going to talk about specific data from the trial. It's still coming in. It's still being analyzed. I know from discussions that I've had that there's quite a lot of insights coming already from changes in the cancer cells, changes in the tumor microenvironment, changes that lead to hypotheses about what we could do differently, what we could do in addition to KRAS inhibition. Um, as I said, I think for me at least, this does provide a pathway. It isn't for everybody. And let me emphasize your point. Drug companies will always focus on the clinical endpoints in terms of getting the drug approved. Do you see response? What percentage of patients respond? What's the duration of that response? That's what the FDA is looking for. The FDA is not asking you to do kind of deep science that we're talking about today. But what the deep science does is allow you, and frankly speaking, the field, to leapfrog the next step.
Right.
Unfortunately, we know that resistance will occur, not necessarily in every patient, but in many patients. That's been a lesson learned over now many years with this type of drug targeting this type of pathway. We could wait until that resistance happens and ultimately study that, but the goal of this project that we're doing together is to get a head start, to understand that information as soon as possible so that we
Yeah.
can design the next best therapy much faster, maybe saving years off the development life cycle. So that's how I see it. And it sounds like you see it similarly.
Very much so. This is all about the future and what comes next. And frankly, the future is coming faster than ever. We already, many patients have been treated with daraxonrasib over the years. More of them now being treated on the expanded access program and then ultimately commercially. And most of those patients will relapse. It's not yet a curative regimen in most contexts. And so we have a lot more work to do. That's why, our emotions, back to your earlier question, we're very deeply emotional and proud about what's happened, but it's more about what has to happen next.
Yeah.
And so this is fundamentally important to moving the needle for the next innovation.
Yeah.
So we're deeply committed to it and very, very happy to be part of it.
Yeah. So again, thank you for that partnership. I'm super excited. I don't know how long it's going to be before we'll start talking about the results, but when we start talking about the results, I know there's going to be a lot of excitement around them. Mark, I want to switch gears slightly. I know that pancreas cancer is a focus for you as a company, but it's not the sole focus for you. Talk a bit more about other disease indications, other RAS-driven cancers. Where is the company beyond pancreas cancer? What are you thinking?
RAS is really the most common genetic driver of human cancers. And not only are, most pancreatic cancer, I think the number, by the way, is much more than 90%. We just quantitate it based on databases, but there's a lot of significant negative false negative testing rate in pancreatic cancer. So I think the number's higher. And even for most of the tumors that don't have a RAS mutation, they have something else that's driving signaling through RAS. Colorectal cancer, 50% carry a RAS mutation, but the other 50% respond to an EGFR antagonist, and EGFR signals through RAS. So wild type RAS is a very important player in the other half of colorectal cancer. And in lung cancer, 30% is driven by a RAS mutation, but there's another 30% that are driven by other mutations that cause signaling through RAS. So maybe two-thirds of all cancer. So of that cancer type. So it's a very common driver. We are interested in all of the major histologic types that I just mentioned, major epithelial cancers. We're interested in other tumor types, biliary cancer, and so on. And some of these are relatively small indications, but we're finding ways to get to those too. And we're interested in all RAS drivers, whether they're mutant or even non-mutant drivers. We have an extensive pipeline. We now have 4 compounds in the clinic, all of which are RAS(ON) inhibitors. We're very excited about all of those. And then we've got a whole new, essentially, generation of innovation coming that came out of laboratory work, that virtuous cycle I alluded to earlier, that are catalytic, have catalytic activity based on hydrolysis of GTP. And I think we'll keep innovating. So we're here for the long run. And maybe to tie back to the previous question, I'd say if you're interested in the disease and not just in your product, you have to invest in the underlying science. If you want to be in that disease and making a difference for the long run, you have to invest in it, and you can't wait.
Yeah. And I think although the different cancers are different and the specifics do certainly matter, there will be common lessons learned across the RAS-driven cancers that will inform each other. So as you do studies in lung, you'll learn about pancreas and so forth. I've got some questions coming in from the audience, Mark, and I want to get to some of them. We talked a bit about response and resistance, and one person is wondering, have you learned already about resistance mechanisms? I'm not speaking specifically about the Breakthrough Cancer Lustgarten collaboration per se, but more broadly, are you beginning to learn about how resistant cancer cells emerge?
We are. The main tool that we have is circulating tumor DNA, because biopsies are hard to get, serial biopsies for patients. And so typically, if somebody relapses, you can just hope that you can get a blood sample, find some circulating tumor DNA, and sequence it. And from that, we have a pretty clear picture that most resistance to RAS inhibitors is driven by the tumor finding some way to reactivate RAS despite the inhibitor. It's a really important point, and I'm going to take us back to the 1990s when the term oncogene addiction, first came out. I think it originally meant that if you turned on the oncogene, the tumor grew, and if you turned it off, the tumor stopped. And that's what Dean Felsher did some wonderful work on. But I think there's another element of addiction, which is sort of the more common street language for addiction, which is that if you're addicted, you do everything you can to get the fix. And a tumor cell clearly does that. And I don't mean to anthropomorphize about it. I think it's just through natural selection. But because there's a wide range of things that a normal cell can already do to adapt to its environment, the tumor can take advantage of those. And it can increase signaling that can either overwhelm or work around RAS. So we've seen that. There's a fair amount of evidence now that, for example, in pancreatic cancer, the most common genetic alteration that you see in the presence of daraxonrasib over time is amplification of the mutant allele, which simply floods the cell with even more RAS signaling to overcome the inhibitor, which, by the way, is part of why a catalytic inhibitor could be important in that context. But that's the main thing. There are sometimes some other routes that don't have anything to do with RAS, but they're much rarer. And then there are some tumor cells for which we don't know what the resistance mechanism is because there's nothing genetic. There's not a genetic signature.
Yeah. Relatedly, folks have asked, are you already thinking about combinations to address these or other forms of resistance that you might imagine? So I'll let you answer that. But another question, which isn't exactly the same, is combinations with chemo. It's sort of a natural combo. There are actually scientific reasons why that might be a particularly attractive combination. So talk a bit about your thinking about combination studies, even now as we're learning about the response.
Really, really important. I think that the treatment of RAS diseases will evolve very quickly into being combination strategies, uh, in part to bat down resistance mechanisms, and maybe create greater durability. We already have extensive phase 1, 2 studies underway on a range of combinations, and we now have combination studies in phase 3 trials. I'll just mention a couple of those. Uh, Duraxonrasib itself is now being studied in combination with chemotherapy in first-line pancreatic cancer. That phase 3 trial is now open and running on a global basis. So we'll get an answer to that question. The earlier phase 1 data were quite strong, that the combination was very, very active. So we expect that to go well in a phase 3 trial. We've just announced recently the initiation of zoldonrasib plus chemotherapy. That's our G12D mutant selective inhibitor with chemotherapy. That global phase 3 trial is now underway based on compelling, promising phase 1, 2 data. We also, and I think you alluded to it earlier, are interested in combining two RAS inhibitors with each other, which is kind of a little bit of a mind-blowing, idea. When we first proposed it, people were like, what are you talking about? But there is some logic, and I think this is what you were alluding to, in taking sort of the broad-based daraxonrasib as a broad suppressor of RAS of all types that might emerge in a cell, and adding to it a mutant selective inhibitor that targets the original mutant form that was the driver. And it turns out in preclinical models, and now we can say in humans, that putting those two things together can be very active, even more active than either one alone.
Yeah.
And we've seen this both with a G12C inhibitor plus daraxonrasib, as well as a G12D, the zoldonrasib plus daraxonrasib. And so there is a phase 3 trial that we will be starting with that combination as well. Highly interested. We're also interested in combining with immune access inhibitors. We're interested in combining with other antibodies that are targeted agents like cetuximab, EGFR, and so on. And we have a lot of work going on. So yes, we think combinations are going to prove to be very important, including combinations of targeted agents like with PRMT5 inhibitors, et cetera.
Yeah. Yeah. And I think that the list will increase and increase. And, the study that we talked about, the collaborative study that we're doing together, will nominate additional potential targets.
For sure.
I think in particular, the microenvironment changes that we will likely see, including, as you just alluded to, the immune system. I think there's very good signals in the literature, preclinically. My lab happens to work in this space too. The activation of an immune response in the setting of KRAS inhibition seems clear now. And if you can take advantage of that, you might be able to stimulate the immune system to sort of clean up what's left behind
Yes.
and potentially prevent resistance because there's nothing there to form the basis of that resistance. So I'm optimistic, therefore, about the immune-based approaches as well. I have a question here, and I don't want to put you on the spot, so please feel free to dodge it. Related to expanded access and following the phase 3 data release, I know that the FDA approved you for expanded access for daraxonrasib. Maybe you could explain to the audience the difference between being enrolled on a clinical trial on the one hand, and then the expanded access program, and maybe just say a word about where you're at relative to expanded access.
Sure. It's really important, and we did move very quickly to seek engagement with the FDA. They moved super quickly to clear us to proceed. And within weeks of announcing that program, we had begun treating patients with daraxonrasib. And now there's a very large number of patients who have gained access across the United States at both large and small institutions. So it's a very, very active program serving, we think, a large number of patients. Really, there's a little bit of bureaucracy associated with it because it's an unapproved drug. and it requires sort of a two-key mechanism. We have to approve that the patient goes on the drug because we have to then ship it somewhere, but the doctor who's prescribing it also needs their own institutional approval to participate in the program. And that's led to some variability as to how quickly institutions came online for it. Now there's a very large number of institutions, large and small. Um, and when a patient shows up at that institution and is diagnosed or is prescribed daraxonrasib, and they send that prescription into us digitally, they get evaluated within 48 hours to see if they just meet the criteria, and then we ship out drug. And we've shipped drugs to many institutions on behalf of many patients. Once a patient's on it, they're just on it for free. And the only reporting requirement as compared to a clinical trial where there's extensive reporting and monitoring patients, basically patients are managed by their physician as they would be, and only if there are serious adverse events are they obligated to report it to us so we can share it with the FDA. So we've really streamlined this to make this about patient care. It's not a clinical trial. We just want to get patients on drug if they want to be on it and if they qualify, if they're eligible before the product is approved.
Yeah. We've talked a bit about what's coming next for the company. You've mentioned several additional clinical trials, not only with daraxonrasib, but zoldonrasib and other compounds that you're developing. I know that there's intense interest in, well, what's coming next for daraxonrasib itself. And I've heard you quoted as saying, you're working closely with the FDA and providing information in real time.
Yeah.
Can you say a bit more about where you're at in that process?
Yeah, well, I think we're nearing completion of the submission. It's been a rolling submission, and we're nearing completion. We're very pleased with how things are going. We can't speak to the timeline for the FDA review, but they've had the opportunity to receive the information as we could make it available and have been reviewing all along. So I think we're getting there. On top of that, just this morning, the EMA announced that it had created a new expedited review program called a phased review process. And daraxonrasib has entered that process and has already started its phased review. It's similar to a rolling review, but it's allowed us to begin providing them with information as well, because they're very anxious to make it available for patients in Europe as well. And we've received global engagement from regulatory authorities, and we'll do what we can to help move it along. So I think we're in a good place. It'll be a better place when we hear back from the FDA what they want to do.
Yeah. Well, given what I'm sure is a lot of inbound and a lot of work related to that and the other things that we've talked about, I'm particularly grateful to you for taking the time to talk to us today. The collaboration that we have at Breakthrough Cancer / the Les Gutton Foundation with RevMed, I think is first of its kind in terms of the depth of science that's being performed on patients, the level of data, the quality of those data, the ease with which those data are being shared across the country, across academic medical centers and investigators, between those academic investigators and the company. We at Breakthrough Cancer have invested quite a lot in data infrastructure, data aggregation, data analysis, which is very much part of the recipe for success here as well. I'm super excited about what those data show us, and I think we'll begin to talk about those in the months ahead. In the meantime, Mark, I want to thank you again for taking the time today, and wish you and Revolution Medicines every success with this program and all of your others. You've been a tremendous partner, and I'll give you the last word.
Oh, wow. Well, I want to thank you for your leadership of Breakthrough Cancer and of the field in general, and for helping initiate, drive, and guide our collaboration. And it's really fantastic to have a chance to talk with you about these things here, and I appreciate the invitation.
Wonderful. All right, Mark, thank you. To the audience, thank you for joining us and staying with us for the hour. As I said, this is a rolling series of webinars at Breaking Through. Stay tuned for the next edition, which will be coming up in the weeks ahead. Take care, everybody.